Hi, it's Mr. Dare here. We're thinking about population ecology here, just recording from my study in lockdown, so if there are kids making noise in the background, do forgive me. First of all, when you think about key terms in ecology, and I'm going to give you a moment, I'm going to pause the video here, or you can pause the video here, and I want you to see if you can match the key terms at the bottom with those, with the definitions above it. Have a read through, see what you think. Well, I hope you had enough time to pause and have a go there. Let's flick on and see how you did. Right, first of all, niche. The exact role an organism plays in a habitat, and it can be thought of as an n-dimensional hypervolume. This is an excellent term, and I'm going to put a link up in the top right to Mr. Anderson's video. This is assuming that you are going to watch that video from Bozeman Science. Habitat, the place that an organism lives, that's a pretty easy one. And then the community is all of the different species in a habitat. Environment is those abiotic factors that might be the quantity of light, the light intensity, the precipitation you'd get per meter squared per year, other abiotic factors such as that. The population is a number. That is the number of individuals of a particular species, a particular time, particular place, and that are able to interbreed. And then finally, the ecosystem, that's kind of everything brought in together. That is all the biotic and abiotic factors together and their interactions. So armed with those terms, let's have a think about a few concepts. Number one, what happens to populations? Well, recently, during lockdown, a lot of us have spent a lot of time looking at exponential graphs, looking at growth graphs. And you know what? The growth of COVID-19 has in many ways matched, well, it is mirroring the growth curves of organisms in particular environments. And in its ideal ecological niche, organisms, they reproduce, their numbers increase exponentially. And here, let's go back, spoiler alert. Here, we're just going to focus on this one. Let's have a think about some of the phases. Actually, what you might do, as you introduce bacteria to a particular place, you might indeed have what is called a lag phase. Now, this is when the bacteria are starting to switch on particular genes, which will flick on certain enzymes and so on. So if you're looking at a very short time scale for bacteria in a jar, you're going to have a lag phase before the exponential growth phase. That exponential growth phase is, if I can find my cursor, there it is going on here. And that is called the log phase. And then what you will find actually is that tails off and it comes off like that, and it reaches a maximum. Now that is as resources become more limited, so the population can't carry on growing indefinitely. You can't feed an infinite population, and so bacteria, just as every other organism, will tail off, and that is known as the carrying capacity of a habitat. So whatever organism it is, its habitat will only be able to support a certain number of them. Now, in the wild, in reality, what that will do is that the population will fluctuate around this, and it'll be pretty random, pretty chaotic, but it'll fluctuate around that carrying capacity. Now, actually, if you have bacteria in a jar, there's something else that happens. Not only do you have this flat, stable phase here, but actually the population starts to crash. And that is because during this phase here, the death phase, you have in this jar an accumulation of all the various toxins that have been produced by the bacteria themselves. So their excretory products are going to poison them and their population goes down. But that's not necessarily just true for bacteria. You may get in other populations factors such as disease, which will bring that population down at some point, maybe causing that fluctuation, maybe causing it to crash in the long term as well. So that is the death phase, and particularly the death phase occurs in sealed containers, in fixed environments. Now that's not going to be the case for most organisms because we have a constant supply of energy from the sun. The graph on the right is the same graph. It's the same graph, but to a different scale, to a log scale there. On our log scale, that exponential curve there flattens out to become this straight line. They are the same numbers, just shown in different ways. The log scale flattening out an exponential to make it a straight line. So, using that and applying that, we can then start to think about what happens when a species isn't growing alone in an environment. These are two species of yeast, Paramecium aurelia and Paramecium caudatum. When both of them are grown alone, their growth curve shows the same pattern that we saw for the bacteria. So aurelia, and then levels off up there, that's normal. And then the caudatum, there we go, up this way and along like that. So they both reach their carrying capacity in that particular jar of nutrient broth and level off. However, it becomes a little more interesting when we put the two in together. Now, initially, both of them start to grow. Now, actually, the caudatum manages to pick up growth much faster. Interesting that actually it's faster then than it is just by itself. This competition seems to stimulate it to fast growth. So up it goes, but then it dies off, and whereas the aurelia, that carries on and reaches the same level as the caudatum becomes extinct within that particular nutrient broth jar. So the aurelia reaches the same level it was at previously. This is taken as evidence that no two species can precisely coexist in the same ecological niche. Now, it is simply a model, it is not proof, but it is modelling an environment, an ecosystem, and seeing what happens to those populations. So again, go back to that Bozeman science video, have a look at that, see how that marries into it. Thank you. Moving on, there are other examples of competition. Competition isn't always quite so straightforward. Going back, I mean, these two are competing for the same, exactly the same nutritional supply dissolved in that broth, and one wins, the other doesn't. It's a little less easy to spot it for the red squirrel and the grey squirrel. On the one hand, they're both squirrels, they're both like eating nuts and similar things. But they don't occupy the exact same ecological niche, but it's similar enough that with the introduction of the grey squirrel, I think back in the 1800s, their niche was so similar that the competition had to occur between the two. The grey squirrel a little larger, maybe a little more aggressive as the rumours go. And if you look at this graph here, it's a graph measuring increase and decrease in population. So initially in Wales, for some reason or another, this population of grey squirrel dropped, but then started rising again. And the red squirrel, just with the introduction of the grey squirrel, it dropped. Now it's curious as to why both of them dropped here. And we'll come back to that in a moment. Meanwhile, in Scotland, actually, I have seen plenty of red squirrels in Scotland, and they're still there in reasonable numbers, depending on where you go. The grey squirrel dipping, rising, and so on. You can see there doesn't seem to be an immediate a piece of evidence from these data here that the grey squirrel is outcompeting the red, because the red is rising, the grey is falling. So what this is saying is it's sometimes a more complicated picture. Here you can see that grey squirrel goes up, red squirrel goes down. Does that prove that as the grey squirrel population goes up, red squirrels are outcompeted and pushed out of their ecological niche? No, it's not a proof of it. It's a correlation. And you're going to have to look more carefully at factors before you conclude causality from correlation. And just going back to this one, when both of those populations dipped, maybe there are other factors affecting this. Maybe there's a particularly harsh winter, maybe a particularly dry summer, meaning the plants are not producing enough acorns, or the oak tree is not producing enough acorns. That's unclear. So correlation doesn't equate to causality. Nonetheless, evidence suggests that the greys came, the reds died out in most parts of England. There probably is a causational relationship. So that's red and grey squirrels, and that is an example of interspecific competition. The other word that we need to know is intraspecific competition. That is competition within a species. So how individuals of the same species compete for food, shelter, water, mates, etc. Predation has a big effect on populations, both of predator and prey. Classically, we think of the snowshoe hare and the lynx. This was easy to measure back in the 19th century when fur traders would hunt both of these species for their pelts, and therefore there was an easy way to look back through the records of how many pelts were sold in particular markets to estimate the population size of these species. And we have this classical shape here. The prey goes up, and then when you've got a large amount of prey, so the population of predator follows, and as that rises, well, so the population of prey falls again, and so on. And they kind of follow each other. They mirror with a time lag between each one. Again, it may not be quite so simple as this. Before I say that though, note that the scale on the y-axis is undetermined here, and normally when you see these graphs, the scale for the predator is different to the scale from the prey. You will always have a larger number of prey individuals than predators, otherwise you're going to have a very fast population crash in the predators. Let's have a look at what complicating factors there might be in this, because of course it's not just predation which is going to hold the snowshoe hare population down. There are other factors such as, is the winter particularly cold and harsh that year? Can they get enough food? Is there another biotic factor, maybe a pathogen coming in? And if you look at this graph here, I'm just going to mark it up a little bit so that we can see what's going on. Just marking the tops of the bars. Now, in this experiment, there were three conditions. The experimenters fenced off a 1 kilometre squared area of habitat in Canada. In one of those areas, they added extra food for the hares. In one of those areas, they didn't add the extra food, but they did exclude lynxes. And in the other, third area, they both added food and excluded lynxes. This bar here, the control, which I don't think I need to mark up, is what happened in a control area where they did nothing. So the height of the bars correspond to the relative population of hares, of the population of hares relative to the population of the control regions. And interesting, when they add food by itself, yes, you get a bit of a rise, bit of a bump, and it comes back down, and it's not massively far from the control. Likewise, when you exclude the lynx, it bounces around a little bit, but really it's just there at control level. And it's only when you give both additional food and exclude the lynxes that you get this population explosion of hares. But even then, you can see it goes up, but then comes back down. And there seems to be no real explanation here, included in these data, as to why that population might crash, unless the food added isn't sufficient, or maybe as populations increase, so population density increases. And as population density increases, you get a greater danger of pathogens being able to spread easily within a population and therefore reduce their numbers pretty quickly. Maybe, of course, it was just pretty simple. It was a cold winter and that killed off the hares. Well, in order to come to all these conclusions, or indeed speculations, you need to be able to measure a population. Measuring plants or sessile, that is stationary, sitting animals, is relatively easy. You've probably used the grid quadrat that we see on the right hand side here. On the left hand side you can see a pin quadrat or a point quadrat. And simply it is a bar, maybe of wood, maybe of plastic, and you drop this pin through it sequentially 10 times through these 10 holes as you place it down. And each time there is a pin strike, so a leaf of a plant or something like that hits it, you record that as the presence of that plant there. You have to identify the plant using a species guide. In both of these cases, you could use them for random or systematic sampling to give you an idea of population size. We're doing this, of course, because it is impossible really to count a full population in almost all circumstances, because populations are normally very large numbers, and therefore what we have to do is we take samples and then conclude from those samples, multiplying up to get an overall estimate, and it will only be an estimate of population size. And then what do we do for animals that move around? This is a great way. Mark, release, recapture. It involves capturing your animals in the first place in a humane way. If you capture them in a way that kills them, this is not going to work. So that might be in a humane trap, such as a pitfall trap for crawling insects. Crawly, and down they go into their pitfall trap. Or maybe you've set up a light and box trap for moths. Whatever it is that you've done, you take all your caught individuals and you mark them in some way. That might be carefully spraying them with a UV sensitive powder, or putting a tag on like we have on this jackdaw's foot there, that jackdaw tarsal there. And then you, having marked them, you re-release them, then you recapture them again, and use this index to work out how to do that. And I'm going to put a link to an excellent video on mark, release, recapture that you can watch to explain this to you. So that's all, but I want you to pause on this screen for a little while and have a look at these words and see if you can define them without first going back to your notes. Thanks very much, and I hope that helps.